Okay, cool. I guess we'll just go ahead and get started then. So, hello everyone, and welcome to another exciting edition of the EPA's Computational Toxicology Communities in Practice. I am Sammy Hamp, and I'm a Public Affairs Specialist with the new Center for Computational Toxicology and Exposure. Today we will be hearing from Dr. Ruili Huang, Senior Informatics Scientist in the National Center for Advancing Translational Science, about the NCATS BioPlanet. Now before we get started, I just want to ask everyone to please mute their lines. You can accomplish that by hitting #6 on your phone. If you're having any issues with the audio, please let me know in the chat, and I'll do my best to get those resolved for you. And without any further ado, I'll hand this over to Dr. Huang.
Thanks, Sammy, for the introduction. So today I'll talk about the NCATS BioPlanet. So first I'll start with some background information. Just tell you about how this project came about. Within the Tox21 program, we have been trying to come up with a set of in vitro assays and trying to see if we can use the in vitro cell-based assay data to predict in vivo toxicity. So far, within the Tox21, we have screened about around 70 different cell-based assays. And then using this in vitro assay data, we try to see if we can build models using those in vitro assay data to predict in vivo toxicity. So we tried to build models for a number of different in vivo toxicity endpoints. And then here I'm just showing the performance of those models measured by the area under the ROC curve. So this is the results from the toxin assay activity-based models. And you can see that the performance, ranges from about 0.6 to 0.8. So it's reasonable, but it's not ideal. So another way we can predict in vivo toxicity is that we can actually start directly from chemical structures, just using QSAR approaches. And then we can also predict directly from structure to see if we can predict in vivo toxicity. So here I'm comparing the model results of the assay activity-based model and the structure, basically QSAR models. As you can see that the structure-based models actually performed overall better than the assay activity-based models. And then if you combine the assay data and the structure data, you get even better models. So why don't we why we can get good predictive models of in vivo toxicity using just in vitro assay data? So if we look at the composition of the toxonyl assays that we screened so far, so we can see this very heavy focus on two very large categories, pathway categories. One is the nuclear receptor signaling pathways. That's the majority of the assays. And then the other one is, the second one is the stress response pathway assays. And we also have a number of other types of assays covering just or simple cytotoxicity or some genotox assays and developmental tox assays. And some are just like counter screens. So if you look at the assay coverage of the toxonyl assays, so those are mostly viewed as pathways important for toxicity, or people can call them like toxicity pathways. But if you look at the entire biological response space, so the toxicity pathways or the pathways screened by the Tox21 program is only a fraction of the entire biological pathway space. So there are a large fraction of the biological response space not covered by the Tox21 assays, so such as the drug targets and other targets and pathways. So we think that maybe the less than ideal performance of the models based on assay activity data of in vivo toxicity is probably because of this limited biological space coverage of the current Tox21 assays. So the problem is that if we described any pathway, that could potentially result in toxicity. So the question is that how many assays and what types of assays we need to screen to cover enough toxicity mechanisms to better predict in vivo toxicity. So the solution for this problem we thought about is that we need a comprehensive pathway resource that can represent the entire biological response space. And then we can subsequently develop assays based on the entire pathway space. So we have there are a lot of well-known pathway databases, pathway resources out there already existing, but there are several issues with the existing pathway databases. Most of them are focusing on particular areas of biology, such as some are just focusing on metabolism, metabolic pathways, and others are focusing on signaling pathways. So a very well-known pathway database is CAG. So CAG is mostly focusing on metabolic pathways. And then in some pathway databases, data are computationally generated without any further experimental validation or experimental evidence. And then there are also other integration efforts. So people trying to put data from different pathway databases together. Um, as an integrated resource. But most of those efforts are just simple data combination without any further curation or validation to remove errors or redundancies. And then in those databases, different types of data often just mix together. They don't make any distinction between like pathways or just protein-protein, simple protein-protein interactions. And then most existing pathway databases don't provide any additional annotations. They just give you a simple like gene pathway membership, and they don't put any other annotations on the pathways or the gene components. And then some of the commercial pathway databases are um, pretty comprehensive, but those you need to have a license. So because they are like restricted access, and then the licenses are usually very expensive. So so those are associated with a very high cost. So that's why we need a complete, non-redundant and open source solution, uh, a pathway resource for everybody, for all the researchers out there to do integrated data analysis to have easy access. So that's why we came up with the idea of the NCATS BioPlanet of pathways. So we intended for this to be a comprehensive resource for first, for Tox21 program, we can use that for assay selection and prioritization for better toxicity prediction. And as a general resource, so you can use such a pathway system for gene expression data analysis or other integrated data analysis. So we try to collect data from all just public sources so that we can make the resulting product completely public and free access. And then we did manual curation to ensure data quality. Also try to put detailed annotations on source of the data, species, the biological function process of the pathways, and then also disease toxicity relevance of the pathways, and also if there's any assays available for each pathway to to monitor that pathway. So currently in the BioPlanet database, we have 1,658 curated human pathways. that covers about around 10,000 genes, human genes. And then we also have a public browser available for people to get access to this resource. So here's a link to the website. So this shows the workflow of how we constructed the BioPlanet. So here are the list of source databases that we used to compile the BioPlanet. So some of the data sources I think people are very familiar with, like CAG and Reactome, Wikipathways. As a first step, we first merged all the different pathways from different sources by we use an automatic merging algorithm so to merge those databases by gene component. And then we did manual curation on all the pathways to standardize pathway names, gene names, and edited the gene list for each pathway, and also merged pathways by gene function to remove or reduce redundancy. And then we added annotations to the pathways, including such as literature support, category and function tags on each pathway, and also the disease relevance of the pathway, and also asset availability for the pathway. Finally, we created a diagram, interactive diagram for each pathway. And then so here it just shows a screenshot of the BioPlanet website. So here shows the pathway data sources, different databases that we used, the number of genes in each database, and the number of pathways, and showing the URLs to as you can see that like most of the pathways, they have like different numbers of pathways annotated, and even some of the pathways are pathway sources are even no longer supported, like BioCarta and Inside Nature, Sun Signaling. So the website actually all went away. So they're no longer updated. So here shows is like an overview of the data from the source pathways. So here I'm just showing the kind of the redundancy between the different data sources. As you can see that, from many data sources have like the same pathways. So the similarity between the pathways is like 100%. So they're essentially the same pathways, so completely redundant. And then some other pathways from different sources are very similar, with 80%, 90% similarity. And then if you look at the number of genes distribution between those different pathways, so they actually range from even some pathways only have like one gene, and then some very large pathways have over 100, even like 200 genes. So there's a large discrepancy like between different pathways. So for pathways normally with less than 10 or 5 genes, those mostly are just incomplete, so they don't make sense. So this is why we went through the very detailed manual curation to remove redundancy and then merge pathways by similarity in their gene function, gene component. And then we remove the pathways with too few genes or just merge them into larger pathways based on the biological evidence or biological relevance. So the next step is that we put functional tags or annotations on all the pathways. So we grouped those pathways into several big different biological functions or categories, including major systems like genetic information processing, development, metabolism, cell cycle, signaling, and different disease pathways. And then we also have smaller subcategories under each large area, functional area. So we also try to find assays to see if we have assays to measure each pathway. Those are the assay coverage of all the pathways in the BioPlanet from different assay sources. We looked at mostly, assays if we have assays available in PubChem, and then if we have assays in Tox21 or other NCATS programs. So it seems like, and then if not in any of those categories, if there's a commercial vendor available, um, for an assay for a particular pathway. So PubChem seems to have a pretty good coverage of all the pathways. And then we also have a pretty good coverage from the toxonal assays. It's a little bit over 50%, 60%. So this is just another view of the assay coverage of the BioPlanet pathways. So this is more like a Tox21 assay-centric view, and then not and then this shows the pathways not covered by any available assays, just based on those assay sources. And we also looked at the disease relevance of the pathways. This is using data from Omium. Of all the BioPlanet pathways, like 97% of them are relevant for some type of disease based on the Omium data, and then 3% of the pathways have no disease-relevant genes. And then we look at the coverage, assay coverage of pathways by their disease relevance. So you can see that there's a better assay coverage of the disease pathways compared to the other pathways with no disease relevance genes. So for the disease pathways, we only have 11% with no assay available. But for other pathways, we have 34% of pathways don't have any assay available. So this is another view of the assay coverage of the BioPlanet pathways by different more finer pathway categories. So this also kind of shows the same type of result. So you see there's a very good coverage of disease pathways, the human disease category. So most of those pathways have an assay available. And also similarly, there's a very good coverage, assay coverage of signal transduction signaling pathways. So in comparison, there's not so good a coverage, assay coverage of the metabolic pathways. So here, the category metabolism has, so the blue bar shows that there's a lot of pathways have no assay available. So this shows you the BioPlanet browser, the website. So you can just browse all the pathways. So there's a browsing function. And then you can see here that we have the link to the pathway map, gene components, disease relevance, and those are the category and functional tags. And here you can see if there's an assay available for this particular pathway. So you can browse the pathway database, about planet database by you can select a particular pathway category. So here I'm just showing all the adhesion related pathways. So there's 14 of them in the database, and then you can also search by different toxicity relevant pathways. So here you can see that we don't have a lot of pathways annotated for toxicity relevance, because this is still kind of work in progress. Eventually, the number of toxicity tags, toxicity pathways will grow. And you can also browse by disease. So if you just type disease here, then you can see all the different disease types and categories we have assigned to the different pathways. So if you actually click on the disease link here, then this actually brings you, first, it shows all the different types of diseases available, annotated by OMIUM, relevant to this pathway. So we actually provide the links to the actual, the OMIUM disease type. So those are all clickable. And so enter assay availability. So if you click on assay availability, so for this particular pathway, for the AHR signal transduction pathway, so here we are showing the source of the assay, and then so this is the protocol name or the assay name. So if we have a tox21 assay available, we'll provide the tox21 assay name that we use in the tox21 database. And then here is the assay name because we put the data into PubChem. So here is the assay name in PubChem. So if you click on this link, this will bring you the assay entry in PubChem. And if you click on this link, this will bring you to our Tox21 data website. So here it shows all the Tox21 assays with data available. So you can actually click on any of those assays to download the Tox21 assay data for that particular assay. So you can search by disease, just disease name. So here I'm showing example. So you can search by just enter autism, and then you can see all the pathways related to autism. And then so you can actually project all the pathways from your search to the BioPlanet, this blue sphere. So this is why we're calling the database the BioPlanet, because we're using this blue globe or blue sphere. So here on the blue globe, on the BioPlanet, every little dot here, white dot here is represents a pathway. So from this view, you can actually see the relationship between different pathways from your search. So you can see actually a very pretty nice grouping of all the autism related pathways. So they kind of form a cluster on the BioPlanet. Uh, so here I'm just showing you can do a just a general keyword search on the website. So here I'm just showing example. You can search for hypoxia and P53. So when you start typing in the search box, we have this autocomplete function to help you guide you, give you some hints on your search. This actually shows you the search results on the pathways that containing both p53 and hypoxia as keywords, and also shows you the pathways containing either hypoxia or p53. So if you click on the PubChem link, so this shows you all the PubChem assays available for this particular pathway, and then those things will bring you to the actual PubChem assay entries. So if you click on the pathway map tag, you see the diagram, the interactive diagram for this pathway. And then if you click on the genes, you see the list of genes involved in this pathway. So the pathway diagram, so this is an example of a pathway diagram. So this is actually interactive. So you can search for any of the genes that you're interested in this diagram if you have difficulties, locating the genes. So there's a text search box you can enter so for example, you want to see where AHR is within this pathway diagram, so you can type in AHR, and then so this AHR, the node will actually be highlighted, so you can see where it's located. And then we also have there's a legend for the pathway diagram, so you can see what all the symbols are representing. We also provided links to outside resources for some of the components within the pathway. So if there's an entry gene entry for a component in the pathway, then we'll provide an outside link to the entry gene entry. We also have so I don't have an example here, but like we also provide like so if a gene is annotated in the Go term, we'll also give you a link to the actual Go term. So using the BioPlanet, you can also do gene enrichment analysis. If you have a list of genes from your gene expression or RADO-seq, RNA-seq experiment, you can copy and paste your list of genes. You can use either gene IDs or gene symbols into this enrichment analysis box, and then you can specify the p-value cutoff for your analysis and just click on Analyze, and then you get all the pathways enriched in your gene list, your particular p-value cutoff. And then here it shows you the search results, and we also listing the actual p-value um, for each pathway. This is sorted by small to large p-values. And again, you can project your searching results, the resulting set of pathways onto the BioPlanet and see the relationships between the pathways from your gene set. We also provided a tab for all the pathways with currently with no assay available, and then so if other people or anybody is interested in developing an assay for any of those pathways, so you can so they can come here and see which pathways don't have an assay available yet, so they can go ahead and maybe develop an assay for that pathway. Here just shows the pathways with no assay available on the BioPlanet. So it seems like they don't have a specific clustering, so they're all different sorts of different types of pathways that don't have an assay available yet. So we also provided a batch download function on the BioPlanet website. So if you click on the download button, then you see this pop-up. So we provide three different types of data. One is the all the gene sets within each pathway, so all the pathway definitions. And then the second file gives you all the category annotations, the functional annotations for each pathway. And we also provide a very large BioPax file that you can use to generate your own pathway diagrams. As I mentioned before, one application for the BioPlanet for this pathway database is that we can use this pathway resource to develop a optimal set of assays for human in vivo toxicity prediction. So here I'm just showing that we try to so this is kind of proof of concept. We try to first build models for actually this is for human adverse drug effect. We build models using again the Tox21 assay data, and we also added some other drug target annotations. So DTA here stands for drug target annotation. So we added those information kind of as surrogates for other targets or pathways. So when we added those actual targets, drug target information, on top of the assay data, we actually get a very significant improvement in our predictive performance for in vivo human toxicity as represented by drug adverse effects. So you see a huge improvement in assay in model performance. So more interesting is that here we actually used around 2,000 different drug targets and pathways. So interesting thing is that if we just select a subset of those targets, so this is using a set of just about 50, 60 different targets. And then added those targets to the Tox21 assay data, we also see a very significant improvement of our model performance. So this means that so here I'm showing the biological response space again. So we have the TOX21 phase 2 assays and then the drug target space. So um, if we select just a subset of those targets and pathways from this space, if the selected set of pathways and targets are diverse enough to represent the entire biological response space, so I'm using like a different color for different type of targets. So if we have some representation um, for each type of pathways and targets, and then we can just design assays, in vitro assays, to just screen and test those targets. And then we can build models just using those selected assays and targets and pathways. And then we can get a hopefully get a pretty good prediction of in vivo human in vivo toxicity. So we have worked this into a TOX21 cross partner project. We're trying to actually screen, actually have selected assays to screen those additional targets. And then later on, we'll try to build models using the expanded data to see where if we can actually get better prediction of human in vivo toxicity. So to how to so because the you know the pathway space is pretty large, so how we can select just a minimal set of targets, because it's kind of impractical to develop and screen assays for like 2,000 different pathways. So we try to figure out a way to just find a minimum set of targets or genes. So we tried this approach, and then we found that about around like 300 to 400 genes. So those genes participate in each of them participate in multiple pathways, so that we can use just this minimal set to actually cover the entire all the pathways in the BioPlanet. So this just shows the comparing the coverage of pathway coverage of our minimum gene set and the rest of genes. So you can see there's a larger number of pathways covered by our minimal set of genes compared to the other genes. And so this also shows that the minimum set of genes each actually cover a lot of different pathways. So some genes even cover over 100 pathways. So those are kind of like the hub genes or master regulator genes that participate in a lot of different biological processes. So this shows that the assay coverage, assay availability for our max coverage genes. So for some of those genes, we still don't have an assay available. So it will be uh, helpful if someone can develop an assay for one of those max coverage genes. So then we can have this optimal set of assay panel to screen for to generate data to get better predictions of in vivo toxicity. So this work has been recently published in the journal Frontiers in Pharmacology. So here is the citation, and if you're interested, you can go in and take a look at the paper. So going forward, we actually intend for the BioPlanet to be a tool for integrated chemical genomics analysis. So for example, if you have particular disease that you're interested in or a type of toxicity, so to compare the normal state with the disease state, and then if you have gene expression data, protein expression data, and then you can enter those data into this BioPlanet tool, and then get disease relevant pathways relevant for your disease or pathways relevant for your particular toxicity endpoint, and then get targets and biomarkers for the disease or toxicity type, and then develop assays and get data, get small molecule probes. So this whole process is actually reversible, and you can actually, once you have the targets and biomarkers, you can generate more data. And then you can use your data and then the pathways from the BioPlanet to get some hypothesis on the mechanism of your disease or mechanism of the toxicity. So moving forward, so we want to be able to periodically update the pathway content of the BioPlanet database, and then add additional annotations. Like I mentioned before, we don't have a lot of toxicity pathway annotations, so we want to add additional toxicity annotations for like different organ type like liver, kidney, heart related toxicity, and also add drug target annotation information into the database. And next step is we want to be able to add small molecule data and gene or protein expression data into the pathway database for easy analysis. So currently the BioPlanet only has human pathways. So in the future, we want to be able to add the pathways from other species like rat and mouse, zebrafish, all the, you know, uh, often frequently used model organisms. So at last, I want to acknowledge our BioPlanet team. We have a number of people from NCATS, and also so some people from Rancho Biosciences who help us with the manual curation of the data in the database. And we also welcome any feedback from you, so you can just send me email directly if you have any feedback or suggestions. I think that's all. Thank you, and I can answer any questions. Um, yeah, so
Hey,
the first
Ruili, it
question
looks like we
is
have a
what
couple
about
questions in the
genes
chat. Can
that
you
are
see those there?
specific for a particular pathway? Uh, I actually don't. I'm not sure what. So this is from Chris Wee. I'm not sure what they mean by what the question is about. So I think in the pathway database we list all the genes uh specific. Oh, so genes, so they probably mean that genes don't participate in multiple pathways. So genes just participate in a single pathway. Why we try to find hub genes is that, so this is just kind of a hypothesis that needs further experimental validation. So the reason why we wanted to find just hub genes, so is we want to minimize the assays that we need to screen. We want to minimize the number of targets that we need to test to cover as many pathways as possible. So that's why we selected the genes that can cover a lot of pathways. So, this still needs experimental validation. That's why. So we're just doing this like a, uh, kind of like a hypothesis. So we'll try to, we want to try to find those genes first, and then develop assays and screen those assays and see if we can actually get using the assay data to get a better prediction of toxicity. So if that works well, then the hypothesis is like approved. But if not, maybe I think it's a good suggestion that maybe we can find some genes that just only participate in some particular pathways and see if that helps with the with the prediction at least for toxicity. We actually, so the question, I see a question is that, so we cover multiple pathways, but have no idea which. So we actually know which pathways each gene is participating in. So we do have a, yeah, we do know the answer for that. Oh, so the question is about what we mean by assay coverage of a pathway.
And
So
then
far,
below
what
that, it looks like
we
we have
did
one
is
from
that
Maurice.
so if assay is measuring any genes in the pathway, we say that the pathway is covered by this assay. This is not necessarily true. So that's also something that needs further experimental validation. Or if NASA is actually measuring the readout is is measuring an entire pathway, for example, like the P53 pathway assay. So it actually measures the entire pathway. But in some other cases, the assay only measures one or two particular genes in the pathway. But we counted all of those as the pathway is is covered by that assay. Yeah, so that's about the pathways, genes, proteins, with adverse outcome path. Yeah,
Okay.
yeah, absolutely. I
And then
think
it looks
that's a
like we
very good
have
suggestion.
another question right below
So yeah,
that as well.
we can try to add those annotations, about adverse pathways. So that's the next step, more toxicity related pathways and adverse outcome pathway annotations. Yeah, that's a very good suggestion.
Okay. And do we have any questions in the room here?
Let's see.
Okay, I'm
Pathogen
getting a lot of blank stares
modular
on that, but it looks like
pathogens
we have
involve
one from David Gerhold in the
multi-localized
chat.
different cell types. Yeah, so I think that can be handled by, adding more annotation. Like we are also planning because right now we have only, human pathways and human genes. Moving forward, we'll have like, from other, genes and pathways from other species. And then I think we'll just, annotate all the genes and pathways by their, cell type, cell type species. Um, yeah, just I think then just to try to add more annotations as they come.
Looks like Maurice is typing.
Oh, about the so I can first answer the question about the pathway categorization. Um, so we actually used a few
Okay, well, yeah, looks like we got another question from, yeah.
different sources. Um, some of the pathways have some categorization information, and then we use that information. We also used there's something called Go Slim. So Go has some categorization, tags, annotations, and then we also use that. So we try to see, which Go Slim terms are assigned to some, any genes in the pathway, and then we'll use the Go Slim term to annotate the pathway. So this is actually a lot of manual work. So we have, PhD scientists working on, manually looking at each pathway and then trying to decide what category or function tag best describes that pathway. So this is a lot of manual work. The next question is to cross-link the human and animal pathways to identify areas that are in common. Yes, yeah, the answer is yes. That's also, I think that's also a very good suggestion. The last question is the compared path with the string database. We haven't done that yet. But yeah, I think that's also a good suggestion that we can do. We haven't looked at the string database. Hi, Tom.
Okay, we got a question in the room here.
Ruili, this is Tom.
Oh yes,
Is
yes.
the
You can
visualization of the
rotate.
planet itself three-dimensional?
It's
And
a
so
3D, and then you
how do you
can
look at
rotate and you
clusters
can
of
zoom in and
genes
zoom
that
out
might be on
and move
different
it
sides of
around.
the planet? Can you rotate that?
We got some feedback in the chat that people might not have heard that question. What it was is, so I guess you were asking if it's three-dimensional, can you rotate it to compare? Yeah, can you, okay, cool.
I don't know if you can still see my screen, but I'm just showing that how you can rotate the the globe, and you can zoom in or out. Yeah.
Yep. It looks like we got someone typing right now, and if there's not anything else in the room, I think it strikes me that we could probably just wrap it up shortly after that, give everyone a little bit of their time back.
This is about technical, so
Okay, yeah,
I'm not
one more.
sure.
We got one more
So this
question
is
in
from
chat.
Maurice. I'm not sure what you mean by technical complexity, like on the assay side or computational side. Oh, reduce the number of assays. We will probably just take the, just take